talk about one such plasmid. So the features of plasmids which we have read, first feature was circular extrachromosomal DNA. They are circular extrachromosomal DNA. And what they have is they are having an unique replication origin. Unique replication origin. So without this particular replication origin, this plasmid will not be able to divide inside the cell. The other thing which they have is they have a selection marker. These are the unique three things which are needed in a plasmid for its survival. Additionally, they have a multiple cloning sites. These multiple cloning sites have several restriction enzyme sites present. And they must have unique restriction site. That means that particular restriction enzyme will be present only one time. One restriction enzyme site will be present only once in the plasmid. Fine. So clear about all these things?
Yes.
Today we will talk about one plasmid, that is pSC101. So what is pSC101? It is a low copy number plasmid derived from Salmonella typhimurium. So from typhoid causing bacteria Salmonella, this particular plasmid has been identified. And it is a low copy number. Low copy number. And this low copy number means it basically has low replication. That is low copies. You have less copies of this plasmid present. And it was one of the first plasmids. But nowadays we prefer high copy number of plasmids. But since it was one of the first plasmids used in recombinant DNA technology, it still is being used. It is important as a cloning vector in molecular biology. So if you look at the structure of pSC101, it is a circular double-stranded DNA approximately 9.4 kb in length. Key features are It has got a origin of replication. It is unique. And it has got a selection, and that is antibiotic resistance gene, and that is tetracycline R. And it has got multiple cloning sites. These sites are filled with restriction enzymes. Filled with restriction endonucleases. And each endonuclease must be present only once. Is present only once. Now let us look at the origin of replication, that is the ORI sign, ORI sequence. They allow the plasmids to replicate within the host. So without the replication origin, the plasmids will not replicate inside the host. It has a low copy number, that is only 5 to 10 copies per cell are present. So only 5 to 10 plasmids are only present, and it minimizes the metabolic burden of the host because it has got genes, and they produce their respective products. So the chromosomes and the metabolic burden is reduced in the host. 5টা থেকে 10টা তৈরি হচ্ছে, তো ওদের কাজটা কম করতে হচ্ছে। তো যদি 100টা তৈরি করতে হতো তাহলে সেলটাকে অনেক বেশি কাজ করতে হতো। সিম্পল অ্যাজ দ্যাট। It has got the next thing is it has got the antibiotic resistance gene. So pSC101, it carries the tetracycline resistance gene. And this enables selection of bacterial cells that have taken the plasmid. So in the medium plus tetracycline, only those bacteria which will grow, only those bacteria will grow which have taken tetracycline resistant plasmid. The others will die, and therefore it is very important. So you are very sure that your bacteria contains the plasmid which may contain your gene of interest, and therefore it is useful in molecular cloning and maintaining plasmid integrity. So this is the plasmid map. You can see this region is the MCS, or multiple cloning site. This is the origin of replication. And this is the tetracycline gene. This is selection marker. And your gene of interest is cloned inside this multiple cloning site. And so the functional features is it is low copy plasmid, it is stable in bacterial cells, it has compatibility with other plasmids, you can transform with other plasmids, and it is useful in long-term expression studies and genome engineering. Provided tetracycline selection is given, this plasmid stays in the host for a long time. This plasmid stays in host for long time. Application, it is used in gene cloning and expression of foreign DNA. It is used in genome engineering and development of bacterial artificial chromosomes. And it is used for making recombinant protein production like recombinant insulin. So what are the challenges and limitations? Low copy number can be a disadvantage when high yields are needed. You need lots of plasmid, but 5 to 10 per cell, you get very less. It has got limited number of restriction enzyme sites in the MCS. And it has got limited number of antibiotic resistance gene for co-expression experiments. So conclusion, it is a valuable tool in molecular biology due to its stability and versatility. Its use in cloning and genome engineering remains significant in both research and industries. Clear?
Yes, sir.
ठीक है आज we stop here. Yeah, please ask questions. दिनाज कुछ बोल रही थी?
सर बोल रहे थे, समझ आ गया.
ओके ओके समझ आ गया. ठीक है, फिर नेक्स्ट डे संडे को भूलना मत.
ओके सर.
ओके.
सर कल भी तो आपका क्लास है ना?
हाँ, कल तो
कल भी
छुट्टी
तो
है.
आपका क्लास है.
कल छुट्टी है.
सर किसके बिहाफ में?
हाँ, कल छुट्टी है. कल छुट्टी तो.
ठीक है सर.
कल छुट्टी तो. हाँ, कल छुट्टी है. कल क्लास नहीं होगा. तब मैं संडे को